Hi and assalamu alaikum everyone. Today we'll be continuing with biological molecules. We will be starting with disaccharides today. If you haven't already watched the monosaccharides video, I would recommend please do so to better understand this topic. Okay, now let's start this. Okay, now disaccharides. Okay, the word di tells me that something related to two. Now what two? Okay, so disaccharides are basically two monosaccharides that are joined together. Okay, they are complex sugars. Why are they complex sugars? Because they can be further hydrolyzed into simple sugars. Okay, what's the meaning of hydrolyzed? Okay, it means to add water, and by adding water you break bonds. Okay, they are usually covalent bonds in this case. Okay, now let's get started with this. Okay, the disaccharides range from carbon 6 to carbon 12s. Okay, we will be focusing on carbon 12 ones. Okay, their formula is C12H22O11. Okay, and as you can see, we have hydrogen and oxygen in the ratio of 2:1. And unlike in monosaccharides, where we used to have, for example, C6H12O6, in which the number of carbons was also equal to the number of oxygen, so we also had a definite carbon to oxygen ratio and carbon to hydrogen ratio. uh, in disaccharides, as you can see, we do not have that. Okay, we do not have a definite carbon to oxygen ratio or a carbon to hydrogen ratio. Okay, now there's examples of disaccharides. We have maltose. Okay, it's made up of two glucose units. We have sucrose, and it's made up of one glucose and one fructose. And we have also lactose, and it's made up of one glucose and one galactose. We have looked into all these monosaccharides in detail. Okay, now let's see the sources of these disaccharides. Okay, let's start with maltose. Maltose is found in germinating seeds, such as barley. Okay, it's found in germinating seed, for example, barley. And sucrose is found in plants. Okay, it's stored in plants and such as sugarcane. Okay, sugarcane. And the lactose is found as the main carbohydrate in milk. Okay, now let's look at maltose. Okay, now maltose is formed from two alpha glucose units. Okay, the forward reaction is condensation, in which a water molecule is removed, and the backward reaction is hydrolysis, and it's a reversible reaction. And the products in the forward reaction we have are maltose and a molecule of water. Now let's look at detail this. Okay, we have this alpha glucose, we have alpha glucose again, two alpha glucose, and they form maltose, and a water molecule is released. Now let's look at the ring structures of these things. Now as you can see, okay, we have an OH in the alpha glucose, this OH. Okay, and we have an OH also below the ring in this another alpha glucose unit at the fourth carbon. Okay, now for maltose to form, these two alpha glucose units should align as shown. Okay, and when they align, okay, what happens is a water molecule is removed. As you can see, a water molecule H from this carbon and OH from the fourth carbon, and this water molecule is removed. Okay, and when this water molecule is removed, we are left with this O. Okay, this O, and this O then connects then connects these two ring structures of alpha glucose, and what ends up happening is that we have a disaccharide. Okay, and according to a definition, a disaccharide is formed by two monosaccharides. Okay, and we formed a disaccharide here, as you can see. This is maltose. Now we give a special name to this bond. Since this bond is formed by two alpha glucose units, okay, then we call it alpha. And since it's formed between carbon 1 and carbon 4, we call it 1 comma 4. And we name this bond as a glycosidic bond. Okay, so this bond here is called an alpha 1,4 glycosidic bond. Okay, now let's look at the formation of sucrose. Sucrose is formed from alpha glucose and beta fructose. We studied both their structures in the monosaccharides, okay, and these are like this, respectively. Um, for sucrose to form, they have to be aligned as shown. Okay, and what happens is the OH from carbon 1, okay, this is carbon 1, and the OH from carbon 2 in the beta fructose, okay, they come close, okay, and a water molecule is removed similar in a similar way to the formation of maltose. Okay, and the water molecule is removed, and what we are left with this oxygen, and this oxygen ends up forming a bond, okay, between both the carbon number 1 of this alpha glucose and carbon number 2 of beta fructose, and it forms a kind of bridge similar to maltose. Okay, and we also call this glycosidic bond. Okay, the specific naming of this bond is not in the syllabus. Okay, so we just call this a glycosidic bond. Okay. Okay, now let's look at reducing and non-reducing sugars. Now we know by definition that a reducing agent, okay, is something that gets oxidized itself and it reduces the other thing. Okay, now all the monosaccharides, we studied them before, and disaccharides are reducing sugars as they can be directly oxidized, except for sucrose. Hence, okay, sucrose acts as an ideal transporting sugar. Okay, why in plants? Okay, why? Because of its inert and soluble nature in water. And it's transported through phloem translocation. We will study this in transport in plants later. Okay, now why only sucrose? Okay, why not maltose, or why not lactose? Okay, why only sucrose is not a reducing sugar? Okay, the answer lies in the structure of sucrose. So let's see that. Okay, so here we have the structure of sucrose. Now there's one major difference in the structure of sucrose and in the structure of other disaccharides or polysaccharides that we'll study later. Okay, let's compare this to other disaccharides. Now in maltose, we had two alpha glucose units joined together. Now they were joined at carbon 1 and carbon 4. Now carbon 1 was the functional group carbon. Okay, it's the functional group carbon for alpha glucose, but carbon 4 is not the functional group carbon. Okay, that means we still have a functional group carbon in maltose that's not bonded. Okay, that's not involved in bonding. Okay, now let's compare this to a lactose. Okay, in lactose we have uh, glucose and galactose. Okay, now there also we still have the one functional group carbon, okay, that's not involved in bonding. So in both these cases they can be easily oxidized. Okay, now what happens in sucrose is that, if you notice, okay, this is alpha glucose, okay, and the functional group for this lies here. Okay, it was carbon 1, okay, it was an aldehyde, and it was aldehyde with this functional group. This was structure. We have the terminating hydrogen here, and we have an R group here. And in this fructose, okay, this is beta fructose, okay, in the fructose generally we had the functional group carbon. It's a ketose sugar, okay, so the functional group carbon was at carbon 2, okay, and the functional group looked like this. Now, do you agree that we have both the functional group carbons involved in bonding? Okay, they are involved in forming the glycosidic bond, so they cannot be oxidized. That's why sucrose is not a reducing agent. Okay, this is a very important point. Okay, this is the reason why sucrose is not a reducing agent, while other disaccharides or polysaccharides or monosaccharides are reducing agent, because they have free, okay, in a sense that it's free functional group carbons. Okay, they are not bonded to not involved in bonding with other molecules. Okay. Okay, so finally let's look at the test for reducing sugars. Okay, now as we all know, it's very common. Okay, it's the Benedict solution test. Sugars. Okay, let's just go over this quickly. Okay, now it's a Benedict solution is a chemical reagent, okay, and for testing reducing sugars. It's a bright blue solution. Okay, it's bright blue solution because it contains copper 2 plus ions, and we all know copper 2 plus ions cause the color of the solution to become blue. Okay, now the reducing sugars, okay, they can react readily with this Benedict solution by gently heating. Okay, gently heating them. And what happens is that they reduce the copper 2 plus ions to copper 1 plus ions. Okay, now as a result, a precipitate is formed and a color change is observed. The color change is usually from blue, that was initial, and to orange. Okay, or it can be also from blue to light green or greenish type. Okay, this all depends on the concentration. It can also depend on the strength of the reducing sugar. Okay, it all depends on this. Okay, as we know that non-reducing sugars will not react with the Benedict solution, hence they will give a negative test result for this. For example, we have sucrose. Okay, that's just studied. Okay, and we can react sucrose in a way. Okay, let's go over that quickly. Okay, if you heat sucrose with a few drops of HCl, okay, that's hydrochloric acid, and you allow it to cool, and then neutralize the solution form. Okay, neutralize with sodium hydrogen carbonate. Okay, sodium hydrogen carbonate. Okay, this hydrolyzes the glycosidic bonds. Basically, it breaks the glycosidic bond. So if you break the glycosidic bond in sucrose, what you end up getting is fructose and glucose. Okay, and they are reducing sugars. Okay, because they're monosaccharides, and they will give positive results. Okay, that is it for today. We'll meet in the next video. Till then, take care. Allah Hafiz.